Hello guys, welcome back to Dr. Hill's Academy. Today we'll be discussing glycogenolysis. That's the splitting of glycogen. Previously we discussed glycogen synthesis. You can check it out or put it in the description below. So watch it before continuing this video. Stay tuned. So glycogenolysis, we have glycogenolysis is derived from the two words glycogen and lysis, right? So we all know glycogen being a polymer of glucose. And then lysis simply means to split. So we have lysis, which means to split. If we pass glycogen through the reaction of glycogenolysis, when we have glycogen here and then it undergoes glycogenolysis reaction, it forms tons and tons of glucose, numerous glucose. Then when the reverse reaction is carried out, so we have the reverse reaction, which is a polymerization of glucose molecules. We have glycogen, right? So this reaction is called glycogenesis. So we discussed glycogenesis in the previous lecture, and today we'll discuss glycogenolysis. So over here we have our GIT, that's our gastrointestinal tract, the GIT. And then when we take in carbohydrates, say we take in polysaccharides, what should we take in? Say starch, for instance. We take in starch, what happens here? Digestion, and then the digested glucose is absorbed into the bloodstream. So we have glucose making its way into the bloodstream. Here in the bloodstream, we have numerous glucose molecules. So what happens to the blood glucose concentration? It increases. If we have a graph here, for instance, of blood glucose concentration against time, you will see that the blood glucose level at this point is high after eating, just immediately after eating, about 9.0 in millimole per liter. So we've seen blood glucose spiking just immediately after eating, and then this blood glucose is stabilized at some point at about an average of 9 millimole per liter. Why? Because there is shunting of glucose. So glucose is being taken up by the various cells. We have glucose over here, taken up by the tissues of the body, the brain and various other cells in the body, right? However, there is still excess glucose, which the body cannot get rid of, right? The body has taken enough for formation of ATP, right? But then this excess glucose then is mopped up by two tissues, the muscle and the liver. So the liver and the muscle mop up the excess glucose, and what do they form? Glycogen. So we have our glycogen molecule over here. So let's say this is our glycogen. It has been formed by the liver and the muscle. So the muscle also form glycogen from glucose, right? So after about 2 hours, let's say 2 hours after you've eaten, that's 2 hours postprandial, that's 2 hours postprandial. What this simply means is 2 hours after you've eaten. What happens? The blood glucose starts going down, right? So the blood glucose starts falling. And this blood glucose is still falling because the tissues are still making use of glucose, right? So it starts falling, and then what happens? The liver senses it. Oh yeah, the blood glucose is falling, and we need to maintain the blood glucose level. What does it do? It now starts breaking up this glycogen. So it starts breaking down this glycogen to form glucose, which is then shunted into the bloodstream. So we have another steady state maintained by the liver. Let's say this is about 7.0 millimole per liter, maintained by the liver. You did not ask me about the muscle. The muscle is so stingy with this glycogen that it hoards this glycogen for muscle contraction. That's muscular contraction. So the muscle is like our politicians, right? So they hoard glycogen and don't use it to form glucose for us, right? And then the liver is like a loving mother. So let's say this was about 2 to 4 hours. So after about 6, 8, 10 hours, what happens? The liver starts sensing that the glycogenolysis is not sufficient to maintain blood glucose. So it now undergoes another reaction known as gluconeogenesis. That's the formation of glucose from new sources like fats, lactic acid and stuff like that. So this is gluconeogenesis. And then the previous one was maintained by glycogenolysis. So today we would see how the liver and the muscle carry out glycogenolysis. The difference is that the muscle breaks down the glycogen to a certain point and then it doesn't push it into the blood, while the liver breaks it down to glucose and then pushes it into the blood, right? So this baby over here and his mother are responsible for glycogenolysis. We'll see how they carry out glycogenolysis. So we have our first enzyme over here. That's our baby GP, right? So this is our baby GP. The name of our baby is GP. And then baby GP loves to eat. So he goes to the terminal ends. That's the non-reducing ends. Always remember that the non-reducing ends are the terminal ends of the chain. So let me label the chains. Let's label the chains. Chain 1, chain 2, 3, 4, 5, 6, 7, 8, 9. These are 9 chains, right? And the non-reducing end, say the non-reducing end of each chain is the terminal end. So the terminal end is the non-reducing end. So these are the non-reducing ends over here. So baby GP loves to eat glucose from the terminal ends or from the non-reducing ends down. All right. Now, how does he do that? He has this saliva. You can notice babies drool a lot, right? So this would represent our drooling saliva. So baby GP is drooling saliva. And the saliva represents our phosphate. So is this phosphate that is used to break the bond in order to release glucose, okay? Now, when phosphate is used to break a bond, it is called phosphorylases or phosphorylases. That's lysis using a phosphate. And baby GP uses this phosphate. What that means is that when he uses a phosphate, there will still be saliva left on the glucose, right? So we'll have a glucose phosphate, but it is added at position 1. Okay, so see what baby GP does. He comes over here, breaks the first bond with a phosphate. So let's say the first bond, he breaks the bond by attaching a phosphate at position 1. The second one breaks by attaching phosphate at position 2. And that is what baby GP continues to do. So you see that he breaks down numerous glucose molecules, forming glucose 1-phosphate. So we would have glucose 1-phosphate eventually, right? And the difference or the trick here is that the phosphate cannot break the bond after the 4th glucosyl unit. So from the branch point, this is a branching point where 2 glucose chains meet. Where 2 glucose chains meet is called a branching point. And from the branching point, you count 4 glucose molecules. So this is 1 glucose molecule, 2 glucose molecule, 3 and 4 glucose molecules. What this means is that beyond the 4th glucose molecules, that's towards the branching point, the saliva or the phosphate cannot break this bond. Okay, so between glucose 3 and 4 from the branching point, the bond is strong. So baby GP breaks all other bonds except that bond. And once he's done with this chain over here, since he cannot go beyond that, he goes to the next chain, right? So you can count it with me. This is glucose, the first glucose molecule from the branch point, right? Second, third, fourth. So what happens? Phosphorylases, 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 and phosphorylases. He goes over to the next chain, that's chain 7, right? So for the chain 7, we will count from the branch point. Here is the branch point for chain 7, not here, over here, because this was another chain. Now this is another chain linking to chain 7 and chain 8 are linked together, and this is their branch point. So from here, we would count how many glucose molecules? 4. So we'll count first glucose unit, second glucose unit, third glucose unit, fourth glucose unit. Baby GP then comes to break this, adding phosphates, break this, adding phosphates. The cycle continues until we have 4 glucosyl units. Okay, so the same thing goes on for each of the chains, right? So you can see that in a nutshell, baby GP, known as glycogen phosphorylase. So this is the name. GP stands for glycogen phosphorylase because it uses phosphorylases to break down glycogen, right? So baby GP breaks down alpha 1 4 glycosidic bond. Always remember that straight chains, we discussed it in the lecture on glycogen synthesis. Straight chains are made up of alpha 1 4 glycosidic bonds, while the branch points are alpha 1 6. So you can see this red over here, these red bonds are alpha 1 6 glycosidic bonds, and then, so let's say this is alpha 1 6 glycosidic bonds, while the blue chains represents alpha 1 4 glycosidic bonds. That's clear, right? So you can notice that baby GP breaks down the alpha 1 4 glycosidic bonds by adding a phosphate at position 1, right? And then until we have 4 glucosyl units left. Can you see the trick? So we have baby GP breaking down alpha 1 4 glycosidic bonds by adding a phosphate at position 1 and continues until we have 4 glucosyl units left. It's pretty cool, right? Eventually, we have glucose 1 phosphates. That's the product. We have glucose 1 phosphates formed, and then we have something else over here called limit dextrin. So these are limit dextrin. Limit dextrin is the molecule that is formed after baby GP has acted. That's after your glycogen phosphorylase. Please don't call it baby GP. After your glycogen phosphorylase has acted on the glycogen. And always remember that you can see this pampas over here. So this is baby GP's diapers. It represents pyridoxal phosphate. So that's the coenzyme for baby GP. So from reaction 1, where glycogen phosphorylase, that's our baby GP, acted on glycogen, we formed a limit dextrin, and then we formed, that is the first thing we formed, and then we formed the second product, which is numerous tons and tons of what? Glucose 1 phosphate. So this is baby GP's mother over here, and then baby GP's mother wants to help baby GP, right? So what she does is that, as you can see, she has a box, and this box, her toolbox, contains a cutter and a glue. I'll tell you what these enzymes represent very soon. So she uses her cutter and then she comes over here. Remember that she wants to act in such a way that she would alter this limit dextrin so that baby GP can come back and continue eating, right? So she comes here and she doesn't want to break the first bond. No. If she breaks the first bond, it's just a glucose molecule. So she'll either eat it or to decompose, right? So she goes to the end of the chain, that's the alpha 1,4 glycosidic bond here, and breaks it, forming a trisaccharide. So she has formed a trisaccharide. That's a 3 glucose molecule. What does she now do? She used the cutter to cut it, right? She will then transfer this trisaccharide onto this chain. So let's say this was chain 8 and this was chain 7 as described previously. So she will transfer the trisaccharide onto chain 7. What do we then form? If you look at the molecule over here, you would see the trisaccharide here. Okay, so she has transferred this trisaccharide to molecule 7. Now we would have the trisaccharide added over here. She'll do the same for this one. So she comes over to chain 6, breaks this bond, that's the alpha 1,4 glycosidic bond, transfers this trisaccharide onto chain 5, right? So we would now have something like this. So you can see that the cutter had an activity known as alpha 1,4 glucosidase. Alpha 1,4 glucosidase activity. So this alpha 1,4 glucosidase activity, right? Because it broke alpha 1,4 bond. Now, the glue was what she used to attach the trisaccharide onto the next chain, right? So the glue is alpha 1,4 transferase activity. So mother has an alpha 1,4 glucosidase activity which she uses to cut the bond, that's the alpha 1,4 bond, forms a trisaccharide, and then uses the alpha 1,4 transferase activity to transfer it onto the next chain. That's the chain that forms a branch with this chain, right? So chain 7 and 8 are linked together at a branch point. So she transfers from 8 to 7, and she has to transfer from 6 to 5, transfer from 4 to 3, and from 1 to 2. Okay, so you can see that she transfers from a chain to the next chain that forms a branch with that chain. So let's bring the cutter and the glue together. We would now form what? The box. So the whole box consists of the cutter and the glue. So we can say by default that the box, mother's box, has a 4 transferase activity. So the name of the enzyme is a 4,4 transferase. Let's just put enzyme. 4,4 transferase enzyme. A 4,4 transferase enzyme is an enzyme that has an alpha 1,4 glucosidase activity and an alpha 1,4 transferase activity. Pretty easy, right? If you check the video on glycogen synthesis, you would see we talked about 4,6 transferase enzyme, but here we are talking about 4,4 transferase enzyme. So let me quickly illustrate what mother does, right? I just described it earlier. These are the other trisaccharides, and then this over here. Mother would transfer this trisaccharide onto this chain. So let's say this is chain 1, 2, 3, 4, 5, 6, 7, 8. So mother transfers the trisaccharide from 1 onto 2, trisaccharide from 3 onto 4, trisaccharide from 6 onto 5 and the rest, just like that, right? And what we have now is this glucose molecule that is attached by an alpha 1 6 glycosidic bond. So this is alpha 1 6 bond. Over here is alpha 1 6 as well. And then we have alpha 1 6 over here. So what mother does is that mother has to eat, right? So the mother has to eat, it's not just the babies eat, but the mothers don't eat so much, they leave so much for their babies. Now the mother goes to the alpha 1 6 bond. That is, remember, that's the terminal alpha 1 6 bond. Remember that she doesn't want to obstruct baby's food, so she goes to this alpha 1 6 bond, that is the last, right, where it's just a glucose molecule at the end. So she goes there to break this bond. Mother is the forbidden fruit. The baby, that's baby GP, glycogen phosphorylase, cannot break alpha 1 6 bond. So the mother has to break the bond. She breaks alpha 1 6 bond over here. Then she breaks this one as well. Does she break this one? No, she doesn't. That is because the alpha 1 6 bond is not the last bond. She has to wait for her baby to eat until there's a last glucose that's attached by alpha 1 6 to the chain, right? So what's the next bond she breaks? Can you help me? Correct, this bond. And then the last one she breaks is this bond, because that is where they have terminal glucose that is attached to by alpha 1 6 bond. So this activity is called what? Alpha 1 6 glucosidase activity. So mother also has alpha 1 6 glucosidase enzyme. You can see that mother has two enzymes. That's 4,4 transferase enzyme and alpha 1 6 glucosidase enzyme. She uses this hand for 4,4 transferase and uses this hand for alpha 1 6 glucosidase. And this is what mother forms. This molecule over here is what mother forms. This was your chain 7, this was chain 5, this was chain 3, this is chain 1. So you can see that chain 2 was transferred onto chain 1. That's the remainder of chain 2 onto chain 1. Remember that was chain 4 onto chain 3, 6 onto 5, and then 8 onto 7. Okay? And she has already taken off the terminal glucose that was bound by alpha 1 6 glycosidic bond. So mother forms this structure over here. What should we call mother? A debranching enzyme. So mother's real name is debranching enzyme. That's pretty cool, right? Because you can see that mother has totally broken off so many branches from 8 to 1, 2, 3, 4. 4 branches, right? So that's why mother is called debranching enzyme. And this enzyme is a bifunctional enzyme. So it's bifunctional, meaning that it has two functional parts, a 4,4 transferase and an alpha 1 6 glucosidase enzyme. So the products formed by the debranching enzyme are this large molecule over here, and then the second one is glucose. Remember that baby formed glucose 1-phosphate, mother formed glucose, though mother formed very few glucose, right? And the glucose can, leave the liver through GLUT2 transporters, all right? So we have GLUT2 transporters, right? So this is the same molecule the branching enzyme formed. It formed the molecule together with glucose, right? So this is the molecule I illustrated over here. Now what happens? Mother has assisted the baby, right? So the baby comes and continues, breaks alpha 1,4 glycosidic bond, right? So it keeps on breaking the alpha 1,4 glycosidic bond by adding phosphates onto position 1, right? Until 4 glucosyl chain units are, or 4 glucosyl units are formed. That's one, let's say 1, 2, 3, 4. So the rest are broken down by phosphorylases, right? Baby comes over here again. This is 1, 2, 3, 4, 1, 2, 3, 4, and then he does the same thing. So we don't need to go over all the details which we described in the previous one. So you just know that phosphates are attached at position 1, right? So it's basically the same thing that baby does. This is our baby GP. That's our glycogen phosphorylase. And this is the molecule he formed. Okay, he has broken this. He just left 1, 2, 3, 4. This is it. This is 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. And then 1, 2, 3, 4. That's pretty cool, right? Because baby has cut off every other stuff by phosphorylases. This is 1, 2, 3, 4. So every other stuff is cut off. And then there are lots of glucose 1 phosphates. Okay. So what comes in now? Mother comes to assist baby again, right? So the same reaction occurs again. So this is what glycogen phosphorylase has formed. And then what comes? The branching enzyme. And then with her 4,4-transferase activity, she cleaves this bond, and then this trisaccharide over here, she attaches it onto this place. 1, 2, 3. If you notice, these 3 chains, we had chain 1, chain 2, 3, and 4. But chains 4, 3, and 1 are all branching from this main chain. Unlike previously, where we would have something like this and then like this. Okay? Where you know that these two are linked and then these two are linked. But in this case, we have something like this. So the 3 of them are linked to this major chain. Okay? So you can see that mother, through the 4,4-transferase activity, that's the alpha-1,4-glucosidase and alpha-1,4-transferase activity, transfers this trisaccharide onto here. Then she also breaks this bond over here, still with the same activity, and transfers this trisaccharide over here. And the last thing the branching enzyme does with its 4,4-transferase enzyme activity is that she comes over here, this last chain, and then breaks off the alpha-1,4-glycosidic bond, transfers this trisaccharide onto the chain over here. So we have 1, 2, and 3. Eventually, we've broken off this trisaccharide and transferred it over here, broken this, transferred it, and then broken this and transferred it. So what do we have left? This terminal glucose, right? And then she does the same thing with which activity, that's which enzyme? The alpha 1,6. That's correct. Glucosidase enzyme activity to take off this glucose, so that she breaks this bond. So she breaks this bond. That's the alpha 1,6 glycosidic bond, right? And then what does she form? The branching enzyme forms a glucose molecule over here, forms another glucose molecule, and another glucose molecule. And these glucose molecules can eventually escape through what? GLUT2. So we have the glucose molecule escaping through GLUT2. Now this is the long chain we formed. I illustrated it over here. So this is a long chain. So the baby comes over, that's the GP, baby GP, comes over, and what does he do? That's the glycogen phosphorylase. He just breaks off everything with a phosphate. So we would eventually have numerous glucose, 1 phosphate. So everything is broken off. And then he added a phosphate to each of them. So he uses the phosphate to break down all these glucose molecules, saying that we've eventually broken down the glycogen to tons and tons of glucose 1 phosphate with little glucose, right? Now we have glucose 1 phosphate. And this glucose 1 phosphate is acted upon by an enzyme, a very super cool enzyme. And this enzyme does something. I'm sure you all would know the name of this enzyme. It transfers the phosphate from position 1, that's the phosphate, the glycogen phosphorylase formed, from position 1 to position 6. So it forms glucose 6 phosphates. What is the name of this enzyme? From our video on glycolysis, I'm sure you would get the name of this enzyme. It's a mutase. So the name of this enzyme is phosphoglucomutase, right? Phosphoglucomutase, PGM. That's the name of this enzyme. So let's say this is a phosphoglucomutase. Pretty cool, right? Now, if this was a muscle cell, remember I told you that muscle is very selfish and it hoards glycogen, right? Like it doesn't form glucose and release it into the blood. So it breaks down glycogen and then uses it for itself. That's for muscular contraction. How does it do that? This glucose 6 phosphate, the muscle decided not to have an enzyme that will break this glucose 6 phosphate. So the glucose 6 phosphate in the muscle then enters which reaction? Glycolysis. So it enters glycolytic reaction, glycolysis, in order to release what? ATP. That is necessary for muscle contraction, right? Now, in the liver, this glucose 6 phosphate can now be acted upon, right? Glucose 6 phosphate can now be acted upon by a wild enzyme. This wild enzyme is over here in the endoplasmic reticulum. So the glucose 6 phosphate will journey its way to the endoplasmic reticulum to meet an enzyme that has been caged. So this wild enzyme, so this is a wild enzyme that has been caged. You can see it's frowning. Now, for the glucose 6 phosphate to enter the endoplasmic reticulum, remember that this is our endoplasmic reticulum over here. It needs a transporter that will transport it from the cytoplasm where glycogen breakdown occurs to where? The endoplasmic reticulum. And within the membrane, this is the membrane of the endoplasmic reticulum over here. So within the membrane of the endoplasmic reticulum, what do we have? We have these transporters. So it's a complex transporter. You can notice that it's something like this. So it has 3 components. The first one would call T1, this one, that is linked to the next one called T2, which is linked to the next one called T3. It's important you know this because there are some diseases that affect each of them, right? So we'll talk about that in the next lecture. Now, this complex of transporter is called something. What's the name of this transporter? It's called glucose 6 phosphate what? Translocase. So it's called glucose 6 phosphate translocase, right? From the word trans to transport. So we have our glucose 6 phosphate entering here, glucose 6 phosphate, through what? The T1. So the T1 is responsible for transporting glucose 6 phosphate. T1 is for glucose 6 phosphate, right? Now, when glucose 6 phosphate enters the endoplasmic reticulum, it meets this enzyme. This enzyme then breaks the glucose 6 phosphate into what? glucose, and inorganic phosphates. That's cool, right? Yeah. The name of this enzyme is called what? A phosphatase. So this enzyme, the wild enzyme, is called glucose. So let's say G, that's for glucose 6 phosphatase. So you know phosphatase means to split, to yank off a phosphate. This phosphate over here is yanked off, right? So the phosphate here is yanked off by this enzyme to form glucose and inorganic phosphate, right? And the glucose is now transported back to the cytoplasm through T2. So we have T2, let me use the blue, T2 for glucose. And as you would expect, the phosphate will be transported through T3, right? So we have T3 for inorganic phosphates. So we have our glucose and inorganic phosphate. What happens to the glucose? The glucose journeys down, makes its way through the GLUT2, which is the glucose transporter found in the liver. So it uses the GLUT2 to be transported out to enter the bloodstream. So the glucose enters where? The bloodstream. Would have lots of glucose in the bloodstream, right? So when you are fasting, the liver undergoes this reaction to push glucose into the bloodstream, right? So as a brief recap, glycogen is stored in the muscle and the liver. Muscle glycogen is responsible for muscular contraction, while liver glycogen helps in maintenance of blood glucose level. So we have the glycogen over here, which is acted upon by a baby GP, known as the glycogen phosphorylase. And what does baby GP do? Baby GP breaks the alpha 1,4 glycosidic bond by adding a phosphate to position 1 until 4 glucosyl units are left, forming a glucose 1 phosphate, that's numerous glucose 1 phosphate, and a limit dextrin. So the mother comes to assist. The mother's name is Debranching enzyme, and then she has two enzymatic activities. So she's a bifunctional enzyme. She has a 4,4 transferase enzyme on this hand and an alpha 1 6 rather glucosidase enzyme on the other hand. So the 4,4 transferase enzyme means that the 4,4 transferase means that there is an alpha 1,4 transferase activity and alpha 1,4 glucosidase activity, right? So the 4 here represents this first 4, and then the other 4 represents the next 4, right? So remember, glucosidase first and then transferase second. So the alpha 1,4 glucosidase activity is used to break off the bond between the first and second glucose from the branch points, right? And then transferring the trisaccharide to the adjacent chain so that the baby GP can continue its activity, right? And then the alpha 1 6 glucosidase activity helps in breaking this alpha 1 6 glycosidic bond, right? To release glucose, which lives through the GLUT2 transporter and then to form a molecule. So this same reaction occurs with baby GP coming over to act on the same molecule that the branching enzyme just finished acting upon, and then after which the branching enzyme now acts, and then baby GP now acts again. So the reaction continues until we have glucose 1 phosphate, numerous glucose 1 phosphate, which is then acted upon by a phosphoglucomutase, which transfers the phosphate from position 1 to position 6, forming a glucose 6 phosphate. This glucose 6 phosphate in the muscle is now used to form ATP through glycolysis, while in the liver, where we are, the glucose 6 phosphate journeys this way into the endoplasmic reticulum through the translocase, right? So we have translocase T1, T2, and T3. So T1 helps in the transport of glucose 6 phosphate into the endoplasmic reticulum so that it will be acted upon by glucose 6 phosphatase. Glucose 6 phosphatase breaks the glucose 6 phosphate to form glucose and inorganic phosphate, and then the glucose now leaves the endoplasmic reticulum through T2, while the phosphate leaves through T3, and then the glucose now leaves the cell into the blood through what? GLUT2 transporters. So guys, we've come to the end of this lecture. Thank you for sticking to the end. Do well to subscribe, like, put your comments below, and share to your friends so they'll benefit from this lecture. Thank you, and see you in the next lecture.